Hello everyone. Today we are going to start the new chapter number 10, that is biotechnology and its application. In this class, we are going to more focus on NCERT session 10.1. In our previous chapter, we got to know that what is biotechnology is, what is this combination of genetic engineering and bioprocess engineering which enable us to make this recombinant DNA technology. Now in this chapter, we will basically deal with what are the application of that biotechnology. So let's go ahead. Disclaimer. So what are the learning objectives of today's class? First, we will compare the different approaches to increase food production. We will see what are the different approaches, like how we can increase our food production, what are the different approaches that has been followed from very few decades, and now what has been changed so that we could have increased the yield of our food production. Second, we will explain the principle and application of plant tissue culture. Then we will define what is genetically modified organisms. Then we will describe the mechanism of action of Bt toxin. So what is going to be the index of today's class? First there will be introduction session, then there will be biotechnological application in agriculture, which is section 10.1 of NCERT. Then we will give a gist of all slides, that will be our summary. Then there will be some NCERT based questions, followed by keynotes, and end with interesting questions that you guys gonna solve yourself. So let's start. Biotechnology involves industrial scale production of biopharmaceuticals and biologicals. What does this means? This means, like in last chapter what we have studied, that how we can alter the genetic material of organism to yield a new product. So now what biotechnology actually involves, or what is actually its major function and what it is majorly involved in, that how we can use that modified organisms on a large scale, on a very large industrial scale, to produce many biopharmaceuticals and biologicals. So next point, what is say, does this production utilize genetically modified microbes, fungi, and animals? So what does this production utilizes? As I told, as we have studied in the last chapter, that genetically modified means the organisms whose genetic material has been altered, that can be used to produce some biopharmaceutical or some biological products on a very large scale, that is industrial scale production. For example, we can use E. coli for insulin production, that we're gonna study in this chapter, ahead. So that is like how we can use this genetically modified E. coli for the production of insulin. And then there are many other microbes also, which is going to other microbes, fungi, plants, and animals also. For example, we will study about bt cotton, and that we will study in today class only, and that we will see how this use of these things has did the has did the betterment for the society and gave us the product which is very useful for the human needs. Now, what are the applications of biotechnology? We can use it in the production of therapeutics, as I have give example that E. coli is used in the production of insulin. Then it can be used for diagnosis of diseases. It can be this can be used as genetically modified crops for agriculture that we're gonna study in today's class only. Then food processing, bioremediation. So what is actually bioremediation? So bioremediation is the process in which we use naturally occurring microbes, or we deliberately give microbes to kill the or to remove the pollutant from the environment. That is what we call bioremediation. So in this also we can use biotechnology. Waste treatment, energy production, all of these are the application of biotechnology. So, what are the research areas of biotechnology? Three critical research area of biotechnology are: first, providing the best catalyst in the form of improved organism, usually a microbe or pure enzyme. So, what actually a catalyst is? A catalyst is a substance which used to make a chemical reaction faster. If a chemical reaction is taking place in 2 days, the catalyst will make it happen in 1 day. So one of the very important critical research area of biotechnology is that we can provide a best catalyst which can be in form of a microbe or pure enzyme, which will make a chemical reaction perform faster, and thus we can have products, we can get the results faster. Then what is the second critical research area of biotechnology? That it create optimal condition through engineering for catalyst to act. Means it will create an environment, it will create a environment that whose each and every condition is optimal for this catalyst to act, and this catalyst to act more efficiently. Downstreaming process technologies to purify the protein organic compound. We have studied the downstream processing very in detail in last chapter, that is biotechnology process and principle, in which we have seen that this downstream processing majorly involves the separation and purification of our protein of interest. So this is also one of the major area of biotechnology in which we provide technologies to purify the protein and organic compound, because more the more the formulation of our protein or organic compound is pure, the better is the result we're gonna get. In this chapter, what we are going to learn: how biotechnology has been applied to enhance the quality of human life. As I told that last chapter what we have studied, and this chapter we are going to apply that how those things, how those genetically modified organism and all those things have been applied to enhance for the betterment of quality of human life. Specifically, we will focus on biotechnology's impact on food production and human health. Our main focus on today's class, or in today's chapter also, will be that how this biotechnology is impacting our food production and human health. So, what are the strategies for increasing food production? So, there are three strategies which we can use to increase our food production. So, let's see what are those strategies. First is the agrochemical based agriculture. So, this agrochemical based agriculture, which is a conventional agriculture only, in which we use chemically synthesized fertilizers or pesticides to improve the yield of our food. Okay, this is the first method. What is the second method? Second method is organic agriculture, in which the fertilizer we use is organic in nature. Third will be the genetically engineered crop based agriculture. That crop who is a crop which is genetically modified, to give better quality, to give better yield, and to increase its and increase and become more efficient in every aspect. So there are three strategies, as I told: conventional agriculture, that is the agrochemical based agriculture. Th- this we used to use in from very last, like, early, which in which we use this chemically synthesized fertilizer and pesticide. Even today also many people use this chemically based fertilizer and pesticides to increase its crop yield and, to protect it from the pest and all. Second is the organic agriculture, which has shown that, like, how organically we can, perform this agriculture thing and all, which is, and even we have, like, heard a lot of time in our house also that, okay, this is organic, it is pure from any synthetic chemicals, and it is good for health. Third is this genetically engineered crop, which we are going to study in today's class more, like, in depth. So these are the three strategies. Let's go ahead. What is green revolution? So green revolution, it actually happened in, like, in mid of 20th century, I think 1960s to 70s, in between. In this time period, agricultural production significantly increased, primarily in developing countries. Developing countries are countries that have been developing. So this revolution aimed to address food shortage and malnutrition. So this green revolution, why it happens? It happens actually in, like, some time between 1960s to 1970s. And why it has been, why it came into the picture? Because it helps in this time period, agriculture production increased very significantly. And what this revolution aimed at, then this revolution, the main theme of this revolution is to address the food shortages that we are facing and malnutrition. Malnutrition means people who is lack of nutrition, who is deficient of nutrition. Means people who are eating food, but that food has no nutrients in them, and that's why they are suffering from malnutrition. So this green revolution has addressed this two problems. First, it has addressed this food shortage problem, because it has, in this period we have seen a significant increase in this agriculture production. And second, it addresses this malnutrition problem, in which people were, initially what happened, people were eating food, but it was so deprived of nutrients that people are still nutrition. So that's why this green revolution came. Now, what are the key factors? So the key factors in green revolution is that it improved the crop variety. It has came that, like, it comes with better crop management practices. And it gave us the application of agrochemicals, that is fertilizers and pesticides. So these are the key factors that has been used in that has this bring this green revolution. First, it has increased this improved crop variety. Second, it has given us some better crop management practices, and it has given us the application of this agrochemical, that how to use fertilizer and pesticide to improve the yield of our crops. So now, what was the significance of green revolution? So because of this green revolution has what it does to us, that it has tripled the food supply. 3 times more food supply are now available to people. But what was the limitation of this green revolution? That even this, after this much, that it has tripled the fruit supply, the yield was still not enough to meet the demand of growing population. So population was growing so fastly that even it has tripled the food supply, yield was not enough to meet the demands of people. Then agrochemicals are often expensive for farmers. These fertilizers, pesticides are very expensive for farmers. And these agrochemicals are also very harmful to environment. We know, we have, like, studied a lot of time from childhood, we are studying that how these pesticides and chemically synthesized fertilizers, they are causing harm to environment, even to people, like people health. Thus, further increase in yield with existing varieties are not possible using conventional breeding. So, what does it lead at last? We have seen that now even more increase in yield with this existing variety is not possible using this conventional breeding, in which we are using this agrochemicals and all. It was not possible. It has tripled the food supply, but as I told, because of the increasing in demand, increasing in the population of people, it was not possible to meet the demand, and yield was not still enough. And as I said, agrochemical is still very expensive for farmers, and it is very harmful for to the environment. And that's why we understood that after this, now there is not going to be increased in yield with this existing variety, and now it is not possible for conventional breeding, so now we have to switch to some other thing. So now there is need for tissue culture over traditional breeding. So they said that now there is need of tissue culture over traditional breeding. So before that, let's see what is the limitation of this traditional breeding techniques. As I said, supply couldn't meet up with the demand. The population has grown so much that whatever the demand of this crops and all, this supply is not enough to meet that demand. This limitation led to an alternative method, that is tissue culture. So now what does this tissue culture actually mean, and what does this actually do? So so this technology, what does it develop to improve crops faster and more efficiently. Using this technique, what we do, we are able to increase the, you know speed of growing these crops, and even these crops are more efficient. So this tissue culture, this has been learned by scientists during 1950s. It involves regeneration of whole plant from explant. Now what is explant? Explant is any part of the plant. So in 1950, scientists has learned that from explant, from any part of the plant, we can regenerate this whole plant. This regeneration needs sterile conditions and special nutrient medium. But what does this regeneration needs? It needs sterile condition, means condition which is free from any contamination of microbes, fungi, anything, and special nutrient medium. So in the media in which it is going to grow, it should have some special nutrients. So let's see what are those. We will see in the upcoming slides what are those special nutrient. So this is the pictorial representation that how from a plant, for example, this is a carrot and this is a genetically modified carrot. Okay, let's see. And from this plant we take a part of the plant. Okay, this part of the plant, we have this explant, whatever this explant, what we call any part of this plant, we took it in the tissue culture. Okay, and we provided it with special nutrient media. Now what will happen, it will first form callus, that is this formation of like cells, then this form into embroid, then embryo, and then this plantlet will come, and then we will see this plantlet will grow into the individual plants. So what are the things which are essential for this tissue culture? Explant, as I told, any part of a plant which can be grown in a test tube. Totipotency. So before knowing totipotency, let's know what is totipotent cell. So totipotent cell is a stem cell. Okay, is a stem cell that has the power, that has the ability to differentiate into any or every type of cell. Means a cell which is a stem cell and which can differentiate into, which can grow and then differentiate into any type of the cell. It can become root, shoot, everything, that cell. So, and what is this totipotency? Ability of a single cell, that is that stem cell explant, what we call, to regenerate into a whole plant, because now it has the tendency to differentiate into any type of cell. So it will differentiate and become a total, regenerate into a total whole new plant. This is the same picture in which we have seen that how a plant part, that is explant, we can take this explant, we can grow it into the nutrient media, into tissue culture. This will form first callus, then form embroid, then embryo, and then this plantlet will come, and this, from this, we again a new individual plant will grow. So what are this nutrient medium requirements? As we told that a special nutrient medium is needed for this explant to grow into a new plant. So what this nutrient medium requirements are? So this, from this part you can answer the NCERT exercise third question also. So this nutrient medium first required carbon source, that is sucrose. Then it requires some inorganic salts, then some vitamins, then some amino acid, and then some growth regulator, plant growth regulator, that could be auxin or cytokinins. So these are the 5 requirements that should be in nutrient medium to for explant to grow. First is carbon source, that carbon source which is source of energy, it could be sucrose. Then there should be some inorganic salts, vitamins, amino acids, growth regulator, that is auxin or cytokinin. Now, what is the application of this tissue culture? From till now, we have learned that what is tissue culture. That how from a any part of the plant that we call explant, we can grow into a whole new individual plant. Okay. So now, what is this application of the tissue culture? So one of the application of tissue culture is micropropagation. Let's see what is micropropagation. By applying tissue culture, it is possible to achieve propagation of large number of plants. This propagation can be achieved in very short duration. So what it is saying actually, by using this tissue culture thing, by using this whole tissue culture thing, we can propagate, means we can achieve a large number of plants. Okay, from like small plant we are saying we can grow a new plant. So you can just think from how many, just from this one only small explant, how many large number of plants we can propagate, we can we can generate. This is what we call micro propagation, and this propagation can be achieved in a very small duration, very small time. Here in this figure we can see that tissue samples scrapped from parent plant, that they have scrapped a tissue, okay, from this parent plant, and this tissue sample placed in agar growth medium containing nutrients and auxin. And then this, what they did, they took this tissues, this small tissues, okay, that is taken from a parent plant, and then they kept it in a agar growth medium which contains nutrient, as I said, and auxin, that plant growth regulator. And these samples develop into tiny plantlets, and now this plantlet planted into compost, and now from this compost the whole number of plants will be generated. So this is how we can achieve this propagation of a large number of plants in a very short duration of time. This is what we call micro propagation. Now, so all the plants which we produce through propagation, because we have taken this tissue from a parent plant only, so all this plant which produced through this propagation are genetically identical, means they are like whole, like their gene, everything. They are genetically whole similar to each other. They're identical to each other. That means the plants are exact clones of the original from which they were grown. Means they are the exact replica of that original plant from which they are grown. Means they will resemble totally to their parent plant. That's why they are called soma clones, clones of somatic cells. So let's see this figure. In this figure, it is shown that there is a source plant. Okay, this, then what we will do, we will take the bulb corn of small source plant, and then we will do this primary treatment and all, then reproduction, multiplication, and during then what happened, there will be shooting and rooting of this plant because we have did the multiplication of this plant. Then this primary hardening of this plant happen, where we will keep it in compost and all this thing, and then there is secondary hardening, and then this will again become same as the source plant. So in short, you have to understand that this you have to understand that through this micro propagation, whatever the plants are produced, how many number of plants are produced that are genetically identical to its parent plant, and that's why they are called soma clones. Soma means they are clones of somatic cells. Somatic cells they are not from germinal cells, they are from somatic cells. That's why they are called soma cells, soma clones, sorry. Now what are the examples from, like, we can do this tissue culture thing, micropropagation thing. Examples are tomato, banana, apple. They have been produced on commercial scale using this method. This is the same figure. This has shown the micropropagation of banana, like how we can take this pulp corn of source plant, and you can using this, doing this all this primary treatment, then reproduction, multiplication will happen, then there will be shooting and rooting, then primary hardening, then secondary hardening, and then they became a source plant. Now, what are the benefits of the micropropagation? This is some from this session you will get the answer of NCERT exercise 2. So, first, recovery of healthy plant from diseased plant. Okay, we can recover the healthy plants from the diseased plant because we can take a part of the plant that is healthy, and then from that plant we can make many healthy plant even if that parent plant was a diseased plant. How it is happened? Using meristem. Meristem here, it is like apical and the axillary meristem is a virus free even in infected plants, because this meristem is a dividing part of the plant. It continuously divides, so it is free from virus even in the infected plant. That's why even a plant which is diseased, we can take this healthy part which is meristem part, which is free from virus even in infected plant, and this meristem part, this apical and axillary stem part, can be used for this micro tissue culture, and we can produce a large number of plants using this through this micropropagation. Meristem can be removed and grown in vitro to produce virus free plants, successfully done for banana, sugarcane, and potato. So this is the same figure in which we have seen that how we can take a tissue sample scrap from the parent plant, and that scrap tissue sample can grow into the whole plantlets and all. Now other benefits. First of all, it is faster than traditional breeding. It can be produced in a large scale large scale, but in short time. It maintains the genetic uniformity because they are soma clones, that they are genetically identical to their parents plant, so there will be a genetic uniformity. This is the same figure. what is the another application of tissue culture? First, we have seen this application, which is the micropropagation, like how from a tissue, from the somatic cell, from this tissue, we can tissue sample, we can produce a large number of the plants, what we call micropropagation. Now the another application of tissue culture is protoplast fusion and somatic hybrids. So let's see what is this protoplast fusion and somatic hybrids. What we do, we isolate single cells from plants. Then we digest this cell wall to make a naked protoplasm. Means what we will do, first we will take single cells from this plant, and we know that plant have cell walls. So first we will digest, means we will remove the cell walls, so that the naked protoplasm, that the only protoplasm without any cell walls, will be available to us. Now the protoplasm from two different plant varieties, each having a desirable characters, are fused. Means we have taken two plants, okay, we have taken single cells from each plant. Then what we did, we removed its cell walls to make the naked protoplasm, to make a protoplasm which do not do not have its cell wall. Now what we do, because these are taken from two different plants, two different plant varieties, each having their own desirable characters, okay, now what we will do, we will fuse the protoplasm of these two different plant varieties. So let's see it from the figure what we did. So see, here are the two plants, okay, we have isolated its cells, then we have removed the cell wall. This is only the protoplasm now present. Now what we will do, we will induce the fusion of this protoplasm, and fusion of this protoplasm, what it will make, it will make somatic cell hybrid. Now this somatic cell hybrid, it stimulates the division and differentiation of hybrid, because we know that we don't talk about that totipotency. So now this somatic cell hybrid will stimulate the now division and differentiation of these hybrids, and now the hybrid leaves will grown in the culture. So this is how this new plants can be generated. This new hybrid leaves have been generated. Produced means. Fused protoplasm is a hybrid of two different somatic cells called somatic hybrids. In micropropagation, we call it somaclones, because it is from the parent plant and it is like genetically identical to the parent plant. But here we are calling it somatic hybrids because it has been formed from the two somatic cells that are from the different sources. That is, it is hybrid of two different somatic cells. That's why we call it somatic hybrids. And now this process is what is this called? This process is known as somatic hybridization. Somatic hybrids can be further grown to form a new plants. So this is the same figure in which what we did. First we took these cells from two different variety of plants. Here it is leaves, okay, then what we have digested its cell wall. Now there we have this their naked protoplasm. Now we will fuse this protoplasm. Now because of this fusion of protoplasm, we made somatic cell hybrid. This somatic cell hybrid has cells have this totipotency in them, and now it will stimulate the division and differentiation of this hybrid, and there will be formation of hybrid leaves. Now this hybrid leaves have the tendency to grow further to a new plant. Example is pomato. So one of the most interesting example is tomato. How tomato is formed? Tomato is formed from tomato and potato. We have fused this protoplasm of tomato and potato and make it a tomato. So why we made this hybrid plant? Because we want the characteristic of both the plants. We want some characters of tomato also and some character of potato also. But unfortunately, what happened? This plant did not have all desired combined characteristic. So it means what we have thought, ki, okay, it will yield like this, it will be like this, it will be like this. It didn't turn out to be that, and that's why this lack of characteristic does not allow scientists to make it for commercial utilization. So here, how it looks: inside it is tomato, outside it is potato. And see, this was how tomato looked like, which was not desired, which did not give us this desired combined characteristic, and that's why it didn't came for commercial utilization. Same they have tried for radish and cabbage also, but it didn't happen. It grown inversely. Like we wanted root of radish and shoot of cabbage, but what happened? We got the shoot of radish and root of cabbage. That's why it, we didn't use it for commercialization, and it didn't succeed. So now, after this, genetically modified organism. What is a genetically modified organism? So the organism whose genes have been altered by manipulation. This is also something we have studied the whole time in the last chapter, that whose organism whose genes have been altered by any manipulation is called genetically modified organism. For example, there are many genetically modified plants, genetically modified bacteria, fungi, and animals also. So here is the example of normal rice and golden rice. So we have modified this thing. Why? Because in golden rice we have modified it in such a way that there is enriched amount of vitamin A in it, whereas the normal rice doesn't have this much efficient vitamin A in it, but golden rice have a lot of enhanced vitamin A in it. What are the benefits of a genetically modified plant? It can make crops more tolerant to abiotic stresses, means it can be like resistant to cold, drought, salt, and heat. It reduces reliance on chemical pesticides. Pest resistant crop means now we don't have to use this chemically synthesized pesticides to protect our crops from this insect and all those things. It helped to reduce post harvest loss, means after this plant all grows, we have many losses, like post harvest losses. It can even reduce that. This is the same figure of normal rice versus the golden rice. Golden rice, the rice who has more amount of vitamin A in it. Now, what is the uses of this genetically modified plant? It has increased the efficiency of mineral usage by plants. So, it increased the efficiency of mineral usage by plants. So what it happens, this genetically modified plants has this ability to, you know, use this mineral from the soil more efficiently. So now what will happen? It will prevent the early exhaustion of soil fertility, because it has efficiently used the mineral. Enhanced nutritional value, as I have said, that golden rice, it has like vitamin enriched golden rice because it has better nutrition quality, it has more amount of vitamin A in it. And this is also something that you can answer in can get your answer for NCERT exercise 11. So now, what is the benefits of genetically modified organism for agriculture and environment? And this, from this session you will get the answer of NCERT exercise number 6. First, there will be maximized yield, means genetically modified crops will make sure that the productivity is better, the yield is better. We have to use reduce amount of fertilizers and chemicals, means genetically modified crops can be engineered for pest and disease resistance, that example we are going to see in next slides. This reduce the dependency on fertilizers and pesticides, help in sustainable farming with minimal environmental harm. What is sustainable farming? So the farming in which we will get the better amount of good yield of crops, but there will be the minimal amount of any environmental harm. So now, along with that, what we can get? We can get the plants without desirable traits. Okay, genetically modified organisms have been used to create tailor-made plants. We can incorporate traits which we want in our plants. These plants can supply alternative resources to industries also. Resources include starch, fuels, and pharmaceuticals. So means these plants can supply alternative results to the alternative resources to the industry. What could be that resources? That resources could include starches, fuels, and even pharmaceuticals. Now, what is this application of biotechnology in agriculture? So, first we can produce pest resistant plants. So, what are these pest resistant plants? So, the plants which is very, you know, the plants who to whom like their pest could not do any harm. So, Bt toxin is produced by bacterium Bacillus thuringiensis. So, there is a bacterium named Bacillus thuringiensis. It produces a toxin, and we name that, and the scientists name that toxin Bt toxin. The Bt toxin gene can be cloned and expressed in plant. So we can clone and express this in plant. We have studied this all this processes of cloning and expressing in host plants in previous chapter. So we can clone and express this Bt toxin gene in plants. Now what will happen if we will express this Bt toxin gene into plant? That we will see in upcoming slides. So this is a bacterium figure of Bacillus thuringiensis, and we can see that it has ribosomes, endosperms, DNA, cell membrane, cell wall. Because it is like a eukaryote, it do not have compartments for all those things. It has been like spread all over. Along with that, it has Bt toxin crystal. Okay, and how this Bt toxin crystal is going to help us, we will see in upcoming slides. So now what this Bt toxin can help us in, this expression, if we will clone and express this Bt toxin gene in plant, so this expression provides resistance to insects, thus creating a biopesticides. So when we will insert, when we clone and express that Bt toxin gene in plant, we have seen that the plant is now resistant to insects. Now it is a biopesticides, means it has been told like resistance to this insects. For example, Bt cotton, Bt corn, rice, tomato, potato, soybean, and many more. This is the same figure of microbes. Now how this creation of Bt cotton plant happens, from here you can answer this exercise, NCERT exercise 7. Specific Bt toxin genes that were, I told that this Bacillus thuringiensis have this Bt toxin genes. We can isolate this gene from Bacillus thuringiensis. Now this toxin genes was then incorporated into several crop plants. First example let's take is cotton. This is an NCERT figure which show that how this this cotton ball first has been destroyed by bollworms, where second is a fully mature cotton ball. So what happened? Now this toxic gene we have incorporated into plant such as cotton. So because we have talked about that we have incorporated that gene into plant. So what are those genes? So source organism we know is Bacillus thuringiensis. Now Bt toxin genes, we call these genes as Cry genes. So it is Cry1AC, Cry2AB, and Cry1AB. This encodes the toxin. This from here you can get your answer of NCERT exercise 7. So yeah, so what is the name of the gene? Okay, so Cry1AC and Cry2AB. This is the cry gene, which is going to encode for this Bt toxin, and type of the affected insect to whom it can make it resistant, that this gene can make from resistant to cotton bollworm insect, whereas the cry1ab make it resistant to corn borer insect. Now, what is the specificity of this gene? Choice of genes depend upon the crops and the target pest. So, how we will isolate this gene? What is going to be our choice, that which gene we wanted? So, this gene depends upon the crop, that the what crop we are taking. Is it cotton? It is potato, tomato, or any other crop, or it is and what is the targeted pest? If the targeted pest is cotton bollworm, we need these two genes. But whereas if the targeted pest is corn borer, we need cry1ab gene. This is because this Bt toxin are, as I said, insect group specific. So this is a picture of cotton bollworm and this is a picture of corn borer. So now, what is the nature of this toxin? First of all, that toxin is produced as crystal. These crystals contain a toxic insecticidal protein. Bt toxin protein exists as inactive protoxins. So, what was the nature of this toxin? Toxin is produced as crystal, which you can see in this figure also. Then, this crystal contain a toxic proteins, the protein which is responsible for killing those insects. And this protein exists as inactive protoxin. It does not, because we can ask now, if this is toxic, then why it is not causing any problem to bacterium itself? Because it is protoxic, it is in inactive form. So now when it comes into active form, so it do come into the active form, it is harmless to animals, okay, as because the Bt toxin is harmless to humans and other animals, because their acidic stomach environment does not activate the protoxin. So to activate this protoxin, we need a alkaline environment, but the human and this other animals have this acidic stomach environment. But insects, if we talk about insect has alkaline pH in their gut, and once it will get inside this insects in their gut, it will activate this protoxin into toxin. And we already know that this Bt toxin gene is like selectively target specific insect, that two genes code for this cotton insect and the other was for corn borer. So it is very this is selectively target specific insects. So now, what are their targets best? First, Lepidopterans. This involves tobacco budworm or armyworm. Coleopterans, it involves beetles. Dipterans are flies and mosquitoes. So these are the different target pests. What is the action of mechanism of Bt toxin? So first, ingestion. So an insect, example bollworm ingests the inactive protoxin from Bt plant. So for example, what an insect did, it ingests this inactive protoxin from Bt plant, and because we know that this insect have a alkaline pH in its gut, this protoxin is going to activate and become toxin, and this toxin is going to kill this insect. So as the second step suggests, that this is conversion of protoxin into toxin, that the Bt toxin proteins exist as an inactive toxin, but because of the alkaline gut of this insect, it will become toxin and kills the big insect. So this is the whole step, like how this protoxin is going to bind to this. Because of this alkaline pH, it is going to activate and bind to the surface of midgut epithelial cells. Then there will be some pore formation because of this pore formation in this epithelial cells. the cell lysis will happen. They will swell first, and then there will be lysis. And lysis of cells, what will do, it eventually disrupt the digestive system of this insect, and the death of the pest will happen. Okay, so the next methods from which we can make our plant resistant to pest is through RNA interference. So, problem associated with nematodes. So, nematodes are the parasitic worms. This it infects plant, animal, and even human beings. So, this is a picture of like one of the worms, that is Meloidogyne incognita. What does it do to human? It infects to the plants. It infects the root of tobacco plants. It causes a great reduction in the yield of the crops. And to prevent that, there is a novel strategy, what we call RNA interference, that is RNAi technology. So this strategy was adopted to prevent infestation. This process was based on the process of RNA interference. So from this part, you can answer this NCERT exercise 10 question number. What it is actually is, it is a natural cellular defense mechanism present in all eukaryote organism. It involves the silencing of specific mRNA. We know that mRNA, from mRNA we are going to mRNA translate into protein, but it is going to silence that specific mRNA. And what is the mechanism of silencing of that mRNA? Silencing happening due to binding of complementary double-strand RNA. This binding of double-stranded RNA to mRNA molecules prevent this translation of this mRNA, and thereby preventing the synthesis of a particular protein. Now, what is the source of double-stranded RNA? First, it could be taken from viruses. Viruses have RNA as their genome. Complementary double-stranded RNA could be taken from infection, could be used. And it could also came from mobile genetic elements called transposomes. It replicate via an RNA intermediate. Now, what is the application of this RNAi, that is interference RNA? First, we can use the transgenic tobacco plant. Nematode specific genes were introduced into the host plants. This gene introduction was done by agrobacterium vectors. This plant called the transgenic plant. As you can see in the figure, that this first plant has already been infected by this nematode, but in second plant we have used this transgenic plant who have this RNAi technology in it, and that's why it has been protected from nematode. So the mechanism of RNAi in transgenic tobacco plant: double-stranded RNA synthesis. The introduced DNA, that is the nematode specific gene, is designed to produce both sense and antisense RNA into host cells. Double-stranded formation will occur then when complementary these two RNA strands join in to form a double-stranded RNA. This is the whole diagrammatic representation of RNAi, how it has been done. Now, how this mRNA silencing nematode happen? The nematode feeds on the root of this transgenic plant. The double-stranded RNA from the plant enter this nematode cells and initiate this RNAi pathway. This double-stranded RNA is processed into small interfering RNAs, that is siRNAs. This siRNA guide a complex, which is called RISC, to silence the specific mRNA of the nematode. The nematode criticals protein cannot be synthesized due to the RNAi, and parasite cannot survive, and the transgenic plant is now protected from the infection. What is the summary of today's class? That food production options include agrochemicals, organic and genetically engineered crops. The Green Revolution boosted yields but faced limits, leading to a new biotechnological approaches. Plant tissue culture enables rapid plant propagation, that is micropropagation, and virus-free plant recovery. Somatic hybridization fuses plant protoplasm to create novel hybrids like pomato. These genetically modified organisms are genetically altered organisms which provides benefits like stress tolerance and pest resistance. The BT crops use a bacterial toxin gene for built-in insect resistance. RNAi creates pest-resistant plant by silencing parasite genes with double-stranded RNA. These are the some NCERT based questions, that what is somatic hybridization and what is this example. So this somatic hybridization, it is a process of fusing isolated protoplasm from two different plant varieties, among them possessing a desirable characteristic. Example is pomato. It is a hybrid of tomato and potato. Thank you.